Trevor Baker, March 1st, 2022. The first case study I examined was case study 1. This patient is a newborn girl who developed lethargy, hypothermia, and apnea after birth. Her blood test showed elevated ammonia that continued to rise. And to understand why this is occurring, I will explain the normal mechanisms of ammonia transfer in the body. Ammonia is a waste product that is created from the breakdown of proteins. The reason that ammonia is converted to urea to be eliminated from the body is because if ammonia concentration becomes too high, it can become toxic to the nervous system. This process starts with transamination. During transamination, an amino nitrogen is transferred to a keto acid. Specifically, glutamate aminotransferase catalyzes the transfer of the amino nitrogen to alpha-ketoglutarate, which forms glutamate. Glutamate is able to undergo oxidative deamination from the enzyme glutamate dehydrogenase to free ammonia in the tissues. Through glutamine synthetase, glutamine can be formed to transfer the ammonia to the liver. Then, for the ammonia to be unloaded and transferred to the urea cycle where it can be disposed of, the enzyme glutaminase is needed, which reacts with glutamine to form ammonia and glutamate. Another amino acid, alanine, is an important part of gluconeogenesis in the fasted state. Alanine is formed by the transamination of pyruvate, and it is then transported to the liver, where it is converted back to pyruvate from the enzyme alanine transaminase. As we've learned from earlier in this course, pyruvate is converted to glucose through gluconeogenesis in the liver. This glucose can then be transported back to the muscle for resynthesis of alanine, allowing the cycle to continue. Therefore, alanine is a non-essential amino acid because it can be produced in the muscle to be transported to the liver to ultimately provide our bodies with energy during the fasted state. After further analysis, the patient's urine orotic levels were elevated. On the other hand, the patient's citrulline levels were within range. This is important information because it allows me to see what enzymes aren't and are affected. Citrulline is produced from both ornithine and carbamoyl phosphate with the healthy enzyme ornithine carbamoyl transferase. Therefore, ornithine carbamoyl transferase is least likely to be impaired. An impairment in this enzyme can raise orotic acid levels, but also decreases citrulline levels, which clearly this is not the case. The enzyme that is most likely deficient or defective is arginine-succinate lyase, which is responsible for producing arginine. When arginine is not produced, urinary orotic acid levels will increase and arginine levels will decrease. To lower the levels of urinary orotic acid and increase the levels of arginine, the amino acid arginine now becomes essential. Because the enzyme responsible for producing this amino acid is deficient, the body needs another way to take it in. Through arginine treatment and supplementation, urinary orotic acid levels will fall and the urea cycle will function properly. That is the end of case study 1. My resources are below. The next case study that I examined was case study 4. In this case study, a patient is suffering from what seems to be megaloblastic anemia. Before we go into her symptoms, I first want to touch on what megaloblastic anemia is. This is a condition where the bone marrow is producing large red blood cells that are immature called megaloblasts due to impaired DNA synthesis. There are two main causes for this condition: deficiency in either vitamin B12 or vitamin B9. After further investigation and research, I have concluded that vitamin B12 deficiency is the probable cause of this patient's presentation. Vitamin B12 deficiency can be caused because of many different reasons. For example, an autoimmune condition called pernicious anemia can cause deficiency due to antibodies to intrinsic factor being produced, which makes B12 unable to be absorbed. Also, damage to the small intestines or dietary insufficiency from a vegan diet can lead to deficiency as well. The reason I believe that vitamin B9 is not the cause of this presentation is because of the symptoms that deal with neurological examination as well as lack of other symptoms. The neurological examination revealed a decreased sensation to light touch and vibration in the lower extremities. This is a condition that is called hypoesthesia and can be caused by multiple things, but the only deficiencies stated include magnesium, calcium, and of course vitamin B12. To add, a deficiency in vitamin B9 usually comes with other symptoms which this patient does not have. These include memory loss, difficulty concentrating, gray hair, and brittle nails. As for iron, another vitamin that causes many of this patient's symptoms, I do not believe that this is the vitamin that is deficient in this case. Iron deficiency can cause a low hematocrit level, but it is not the root cause for megaloblastic anemia. Iron is not the precursor to blood cells and does not cause large immature blood cells to be released from the bone marrow, causing this condition. Iron is only responsible for the production of hemoglobin, which is a part of the red blood cell that carries oxygen. Vitamin B12, on the other hand, has a hand in red blood cell creation through DNA synthesis. Although vitamin B9 is not responsible for this patient's presentation, it still plays a role in DNA synthesis and megaloblastic anemia. Folate donates methyl groups, which is crucial in DNA synthesis. Vitamin B12 plays a role in this reaction by recycling 5-methyl tetrahydrofolate back to tetrahydrofolate, which is important for the production of methionine. No vitamin B12 means no recycling reaction No vitamin B12 means no recycling reaction, causing folate to become trapped in 5-methyl tetrahydrofolate, and no methyl donation can occur. As vitamin B12 continues to be deficient, DNA synthesis becomes disrupted and red blood cells cannot be formed properly. The precursor cells responsible for maturation cannot function and eventually hemolyze, leading to immature cells being released. With tetrahydrofolate being inhibited, purine synthesis also cannot occur. Different derivatives of tetrahydrofolate contribute to carbons that are added into reactions, contributing to purine synthesis. So in summary, without vitamin B12, tetrahydrofolate recycling is inhibited, leading to the blockage of purine synthesis and the overall dysfunction of red blood cell production. There are different tests that can be done to determine the type of vitamin deficiency a patient has, especially when megaloblastic anemia can be caused from either vitamin B12 or vitamin B9 deficiency. For instance, one lab test consists of the test for homocysteine, an amino acid, and methylmalonic acid, MMA, a substance which is created when protein is digested. In someone who is B12 deficient, both of these will be elevated, while only MMA will be elevated in folate deficiency. It's important to distinguish between the two, because if someone who is vitamin B12 deficient only receives folate, then the neurologic damage will not be treated, only the anemia. As for iron, a test to determine the red blood cell size and color, hematocrit level, and hemoglobin levels can be taken. One who is deficient will have a small and pale red blood cells, low levels of hemoglobin, and a low hematocrit percentage. That is the end of my case study for 4, and my resources are below. Thank you.